Hi children, after memorizing the summary of molecular basis of inheritance, let us move to the application of this knowledge for the welfare of human society. There is one mega project which was initiated in 1990 under US Department of Energy and National Institute of Health America or United States. Why there is necessity of this mega project? They found that the human beings are suffering from number of genetic disorders and hereditary disorders. They found that all the human beings are unable to express their genes at adaptive levels. And there was a challenge in front of the scientist to replace the errors or to find the solution for genetic disorders, and challenge was accepted. To know the cause of genetic disease or heritable disease, they should understand the entire DNA in the human body. Thus, Human Genome Project was launched. Under this Human Genome Project, under the directorship of F.H.C. Crick, US Department of Energy and NIH of America, they estimated that in human, the DNA having 3 multiplied by 10 raised to the power of 9 nucleotides, approximately 3 billion nucleotides. And to know each nucleotide, the cost estimated was $3 billion, as each nucleotide sequence is costing for $3. It was a huge amount, but the challenge was accepted. Various nations come forward and the project initiated. There are 2 approaches under this project. One approach is to know the genes which are coding for proteins, and the genes which are interrupting these coding genes, means they are non-coding sets. Considering these 2 approaches means to know the coding sequences in human DNA and to understand what exactly non-coding sequences are. Under these 2 approaches, Human Genome Project was initiated. To know the coding sequences means whatever we want, only those genes we must study. Under this, they started expressed sequence tags. And while learning this, there was a curiosity. What about these non-coding sequences? Then they decided, let us identify the sequence which are also present in these non-coding sequences, and they initiated sequence annotations. So there are 2 methodologies adopted in Human Genome Project. One, to find the coding sequences in human genome, that is expressed sequence tags, means to know the genes as operons, which are cistrons coding for a protein necessary for metabolic activities in human. And other area where sequence annotations. Now annoying is a word will explain the thing which is troubling you, the thing which is increasing your curiosity, which will not allow you to keep quiet. So there is a big group of scientists, they were curious about these non-coding sequences, and under this they started identifying the nucleotide sequences of non-coding genes under sequence annotations. So children, HGP is abbreviated form of Human Genome Project, mega project, because the cost bared for this project was $3 $300 billion. There are two approaches or two methodologies adopted. One is expressed sequence tags and other is sequence annotations. When they started sequencing the nucleotides, they found difficulty in writing the data because data was huge. Number of notebooks, number of pages, if you use and store as a library, but to retrieve that data was a difficulty. So, the people working under Human Genome Project, they took assistance of information technology. And there was a new branch launched. It is bioinformatics. This bioinformatics includes software people. They created the software which will take the data after sequencing the human genome. Thus, the cost was bared, approaches are made under the methodologies, and bioinformatics is ready to store the data. So what were the objectives of Human Genome Project? First objective, to do karyotype analysis, means to identify all the chromosomes and arrange them according to their length. Second objective or a goal, sequence each chromosome. Third, keep this data safe in the software. And there was a committee made because when the project was launched, some people, they created the issues at the social level, at the legal level, and under the ethical level. So a committee is made where people started counseling for the people who started troubling the group of scientists under legal, social, and ethical issues. Thus, the project is launched. While the project was on, the third objective was set, that each chromosome was analyzed for total number of genes, total number of nucleotides, total base pairs, and the tool was ready to create the database under bioinformatics. The provision is made to create the changes in this database, and the last objective was the committee is made to address the people who created the issues at social, legal, and ethical level. While doing this project, what they found was the DNA, when it is extracted from the cell, it was a long DNA molecule, very difficult to study. So, they used enzymes which will cleave the DNA at specific sites, and those enzymes are restriction endonucleases, REN. Basically, these are available in prokaryotes. So they isolated these enzymes, studied the property, and they designed those enzymes by culturing prokaryotic cells. Number of REN, restriction endonucleases, are synthetically prepared and they used as scissors, which will cut the long DNA into fragments. Then these fragments are isolated as per the length by a technique called gel electrophoresis. Now you may be thinking what this cleaving is, what REN is. Okay, why these RENs are cutting the DNA? What is the gel electrophoresis? Okay, all these questions keep in the mind because there is a unit coming up, biotechnology, where we are dealing with all these basic processes. Then you will understand what exactly they did in developing human genome. So, DNA is isolated from the sample cells. The long DNA is fragmented. Fragments are separated according to the length by gel electrophoresis. They are tagged with radioactive probes. This is a Sanger principle who awarded with Nobel Prize. he developed dideoxynucleotide triphosphates, which are radioactively labeled nitrogen bases. When you take these DNA fragments and dip in the solution containing these probes will attach. For example, radioactive thymine will attach to natural adenine of DNA. Radioactive adenine will attach to natural thymine. Thus, each fragment is identified with number of adenine, number of thymine, number of guanine, number of cytosine. Thus, they found the percentage of adenine, percentage of thymine, guanine and cytosine. And thus each chromosome was sequenced and the huge database was made ready. Now what is the outcome of this project? I will take in the next session. Let us stop here and start thinking what exactly they did under Human Genome Project. Thank you. झालं जरा बरं.